Welcome back to the deep dive. I want you to start this journey by zooming out as far as you can, picturing the sheer, dazzling variety of life on Earth.
Mm-hmm.
I mean, we have everything from extremophiles thriving in boiling hot springs or deep-sea vents to, highly specialized complex organisms like us, human beings,
Hmm.
built from trillions of organized cells.
Right.
At first glance, the diversity is it's
Yeah.
overwhelming.
It is. But if you look closer, and I mean really look, beneath all that surface variety, biochemistry reveals this profound, powerful unity.
Okay.
And that's really the core theme of the foundational material we've gathered for this deep dive. Even life forms that seem completely distinct, they share a common molecular logic.
Oh, so?
Think about it. The core chemical transformations you need to convert glucose and oxygen into energy, what we call cellular respiration, they're essentially identical in something as simple as, say, common gut bacteria, E. coli,
Okay.
and in the highly specialized cells in your brain. Same core process.
That is astonishing. That structural and functional similarity. So our mission today is to unpack this unity.
Exactly.
This deep dive is built on a unit exploring these fundamental architectural and functional principles that govern life. And our goal is to connect molecular structure to dynamic biological outcomes, right?
Yes, moving from the atoms and bonds all the way up to these complex cellular communication networks.
The unifying theme will trace through every system, from the smallest molecule to the largest pathway, is this interplay of structure, information, and regulation.
Right. We're going to see how weak forces define strong structures, how DNA stores the blueprint, how proteins execute the work, and crucially, how all of these systems are precisely regulated to maintain cellular balance.
Okay, let's unpack this. We have to start with the absolute basics, the foundational forces that make all this molecular machinery possible.
And the remarkable solvent where it all happens, water.
So the first principle, the one that governs everything else.
Structure dictates function. That's the mantra. In all of biochemistry, in all of molecular biology, it always comes back to that.
Can you give us an example?
Sure. Look at the structure of DNA. Its precise double helical architecture with that complementary base pairing. It's not an accident.
Right.
That structure is what allows it to function as a highly efficient and robust vehicle for storing, transmitting, and even correcting genetic information. The relationship is purposeful.
And what defines those structures are the bonds. Now, we talk a lot about the strong, permanent covalent bonds that link atoms together.
Mm-hmm.
But what holds these massive molecular structures of life together, like a protein folding into its perfect shape, or two strands of DNA recognizing each other, is actually the power of weak bonds.
Specifically non-covalent bonds.
Yeah. So wait, why are the weak ones, more important than the strong permanent ones?
That's a really insightful question. Because the cell needs flexibility, it needs reversibility, not just permanent strength. Covalent bonds require a huge energy input to break or form. But non-covalent bonds, and this includes ionic interactions, hydrogen bonds, and van der Waals interactions, they are individually weak.
Put together.
But their geometry, their strength, their collective specificity, that's what's crucial. They allow these huge structures to assemble rapidly, interact for just a moment, and then disassemble just as easily when regulation says it's time.
So the collection of thousands of these tiny non-covalent forces is what gives a large macromolecule, say an antibody, its immense stability.
And its ability to bind specifically to one target out of millions. Precisely.
Wait,
Let's
so-
break those down a little.
So ionic interactions are pretty straightforward. They're just electrostatic attractions between charged atoms.
Right.
Hydrogen bonds are weaker than covalent bonds, maybe 4 to 20 kilojoules per mole, but they have this crucial tendency toward linearity.
What do you mean linearity? I mean the hydrogen bond donor, the hydrogen atom itself, and the acceptor, they all prefer to lie along a straight line. And that geometric specificity is vital for orienting molecules exactly where they need to be, especially inside an enzyme's active site.
And then there are the ones we tend to forget, van der Waals interactions.
Mm-hmm. These are just transient fluctuations in electron clouds that create momentary dipoles. Individually, they're the smallest contributors, maybe only 2 to 4 kilojoules per mole per pair of atoms.
But you're dealing with thousands of atoms.
Exactly. Remember the scale of the machinery. When two huge macromolecules come together, like a protein binding to its substrate, a vast number of atoms come into van der Waals contact.
So it all adds up.
The summed effect of thousands of these tiny interactions becomes really substantial. It's what defines the final snugness, that perfect fit of this whole complex.
And all of this is taking place in the cell's primary operating theater,
Water.
the solvent of life.
Water's unique properties are absolutely essential. It's highly polar because of its bent structure, which gives it an electrical polarity. Right, the oxygen side is a little negative, the hydrogen sides are a little positive.
And that polarity leads to two critical properties.
First, water is highly cohesive. The molecules interact strongly through these extensive hydrogen bond networks. You can almost picture it as a constantly shifting net, even in liquid form.
And second,
That polarity gives water its incredibly high dielectric constant, about 80.
okay, what's the implication of a high dielectric constant?
It means water can effectively screen and interact with charged molecules, which facilitates those ionic interactions we just talked about. It's a versatile solvent because it can stabilize charged species by insulating them from each other.
Which is why salts and so many biomolecules can dissolve.
Exactly.
Okay, this leads us to maybe the single most important driver in molecular organization, the hydrophobic interaction. You said earlier this isn't a bond itself. So what exactly is the driving force behind it?
It's the tendency of nonpolar groups, like the oily side chains of some amino acids, to cluster together when you put them in an aqueous solution.
Why do they do that?
Because when a nonpolar group is exposed to water, it forces the surrounding water molecules to adopt this highly ordered sort of cage-like structure around it.
Which water doesn't like to do.
Right. This increased order lowers the entropy of the water, and that is thermodynamically unfavorable.
So the nonpolar groups cluster together, not because they love each other,
Not at all.
but because they are minimizing the total surface area exposed to the water. This allows the surrounding water molecules to maximize their own entropy.
To maximize the disorder of the overall system. Exactly. It's a solvent-driven process. And this drive to minimize the disruption of that ordered water network is what powers protein folding, membrane formation, the assembly of so many massive complexes. It defines cellular architecture.
Okay, moving up in complexity. Controlling the cellular environment is non-negotiable, and that means maintaining a stable pH.
DNA stability, for example, is highly sensitive to acid-base reactions outside of a very narrow range.
This is where buffers come in.
Buffer systems resist changes in proton concentration, and we can understand how they work conceptually using the Henderson-Hasselbalch relationship.
Okay.
It shows that a buffer system, a weak acid, and its conjugate base is most effective at resisting pH changes when the solution pH is close to the acid's pK.
So if the pH equals the pK, that means the concentration of the protonated acid and the deprotonated conjugate base are exactly equal, a 1-to-1 ratio.
Yes, and that means the system have equal capacity to soak up any added protons using the conjugate base or to release protons using the weak acid. It's perfectly balanced.
And what if the pH is far away from the pK, say pH 9, but the pK of your buffer, like acetic acid, is 4.75?
Well, at that point, nearly all the acid molecules have already been converted to the conjugate base.
Yeah.
If you try to add more base to the system, the ratio is already so skewed that the buffer has no capacity left to resist the change.
So the lesson here is that the molecular environment, the forces, the solvent, and the precise pH sets the stage and enforces the rules.
It does. But what about the players that actually use those rules to build the machine?
Right. That's where the proteins come in.
If DNA is the master blueprint, proteins are the workhorses, the tools, the machines.
And while DNA only uses 4 building blocks, the nucleotides.
Proteins are constructed from 20 different amino acids. And this chemical complexity, this variety, is what grants proteins their immense range of functions.
Like catalysis, signaling, structural support, movement.
All of it. And that diversity resides entirely in the side chains. So categorizing those side chains is really the key to understanding the structure.
We separate them into 3 groups based on how they interact with that aqueous environment we just talked about.
Right. First, you have the non-polar hydrophobic chains. These are the aliphatic ones like valine, leucine, and isoleucine, and the aromatic ones like phenylalanine and tryptophan.
And because of the hydrophobic effect, these guys tend to cluster together deep in the interior of water-soluble proteins.
Driving the folding process, exactly.
Like second group.
The polar uncharged groups, serine, threonine, tyrosine, asparagine, and glutamine.
And it's a classic cause and effect relationship here, right? The hydroxyl, the OH groups, found in serine, threonine, and tyrosine, make them far more hydrophilic and chemically reactive than their nonpolar counterparts.
And that reactivity is crucial. Those hydroxyl groups are often the targets for phosphorylation.
Which is one of the most common mechanisms for regulating protein activity.
It's like an on-off switch for the protein.
Finally, we have the ionizable side chains.
Histidine, lysine, arginine, these are positive or basic, and aspartic acid and glutamic acid, which are negative or acidic.
And these can donate or expel the protons, making them essential for facilitating reactions, especially in enzyme active sites, and for forming stabilizing ionic bonds on the protein surface.
Once you have the amino acid sequence, which is the protein's primary structure, the whole game changes. The remarkable fact is that proteins spontaneously fold into these elaborate three-dimensional structures.
Determined solely by that primary sequence.
Right, a principle proven by Christian Anfinsen decades ago.
And this folding process is governed by a huge thermodynamic challenge.
It is. The folding is driven by those weak forces we discussed, primarily the favorable gain in water entropy from the hydrophobic effect. But these weak forces have to collectively overcome the immense loss of entropy that happens when an unfolded, flexible polypeptide chain collapses into a single, highly ordered structure.
The folded state is only marginally more stable than the unfolded state.
But it's incredibly specific, and the structure proceeds quickly through distinct levels. The primary sequence determines the secondary structure,
Which are these highly regular local arrangements like alpha helices
stabilized by intrachain hydrogen bonds,
and beta strands.
stabilized by interchain or intersegment hydrogen bonds. You also see things like reverse turns and loops.
And these are often rigid and well-defined, even though they don't have a regular periodic structure.
Right. And the loops often lie on the protein surface and are crucial for interaction with other molecules or regulatory elements.
This final native structure leads to the principle of cooperative folding. What does that mean exactly?
It means the folding transition is often described as all or nothing. So if you try to disrupt the protein, say, by increasing the temperature, the structure doesn't slowly unravel piece by piece.
So a little disruption causes a big problem.
Disruption in one part quickly destabilizes the entire structure, leading to a complete rapid unraveling, an all-or-nothing transition.
So if we look at a solution that is midway through this transition,
You don't find partly folded molecules.
you find a 50-50 mixture of fully folded and fully unfolded molecules.
Exactly. Now, this suggests a simple two-state existence, but of course there have to be kinetic intermediates. Capturing those fleeting structures is one of the hardest challenges in modern structural biology.
But there's an emerging idea that challenges this paradigm of a single fixed protein structure.
Yes, metamorphic protein.
Okay, this is fascinating.
For most of history, we just assumed one sequence equals one stable structure. But metamorphic proteins exist in an ensemble of structures, often two distinct, mutually exclusive stable conformations, just poised in equilibrium.
So a single sequence, two completely different 3D shapes, and two completely different functions.
That's it. Take lymphotactin. It's a chemokine, part of the immune system. In its standard structure, it's a three-stranded beta sheet and a terminal helix.
And in that shape, what does it do?
It binds to its specific immune cell receptor, signaling an immune response.
But wait, what's its second life?
The alternative structure is a dimer composed entirely of beta sheets. In this completely different conformation, it binds to complex carbohydrates called glycosaminoglycans on the cell surface.
And the two functions are mutually exclusive.
They are, but the protein requires both functions receptor activation and cell surface adhesion to achieve its full biological goal.
That effectively expands the functional capacity of the genome without adding a new gene. I mean, the implications of that are huge.
They are. And tragically, when this highly tuned folding process goes wrong, we see devastating pathology, typically in the neurodegenerative diseases.
Right.
In prion diseases, the normal cellular protein, PRPC, which is rich in alpha helices, it switches conformation to PRPSC.
And that new form.
That PRPSC form is rich in beta strands. It forms these insoluble infectious aggregates, and it's capable of transferring the disease by inducing misfolding in healthy copies of the protein.
This shift from a flexible alpha helix-rich structure to a rigid beta strand-rich structure seems to be the molecular signature of misfolding.
It is. We see a similar structural pathology in Alzheimer's disease, where the A-beta polypeptide fragments form amyloid plaques.
And those plaques are.
They are dense with extended parallel beta sheet structures, which are just inherently prone to aggregation. The connection between a tiny structural error and catastrophic neurological impact is profound.
So to understand these fantastically complex, shape-shifting proteins, we need powerful tools. After you get the complete DNA sequence, the genome, the next step is to define the proteome.
Right, and defining the proteome is a massive step up in complexity.
Why is that?
Because the proteome represents a far more complex level of information than the genome. It's not just a list of the possible proteins.
It's what's actually there.
It's the complete set of proteins being expressed, including their specific types, their functions, any post-translational modifications, their location within the cell, and the complex interactions between them, all within a specific biological state.
This complexity means you have to start with purification.
You do. You have to separate them sequentially based on their distinct physical properties. Let's start with size.
Okay.
You can separate by size using techniques like size exclusion chromatography, where small molecules get trapped in porous beads and elute slowly, while large molecules just pass right through.
Or ultracentrifugation.
Right. This quantifies particle movement using the sedimentation coefficient.
And the shape of the molecule matters here, which is an important insight.
It is. For example, the muscle protein tropomyosin, which is very elongated and rod-like, it sediments slower than you'd expect based purely on its mass.
Why?
Simply because its non-globular shape increases its frictional resistance as it spins.
Okay. Next up is separation by charge.
Typically using ion exchange chromatography. This exploits the difference in the net electrical charge of proteins at a given pH.
So if a protein has a net positive charge from all its lysine and arginine residues,
It will bind tightly to a column matrix that contains negatively charged groups. You can separate it from neutral or negatively charged proteins and then elute it by raising the salt concentration or changing the pH.
but the most precise method uses biological recognition, right? Affinity chromatography.
This is truly specific. It exploits a protein's natural tendency to bind specifically to a certain small molecule or ligand.
So if you want to purify concanavalin A, which is a protein that binds to glucose,
You immobilize glucose onto the chromatography beads.
the concanavalin A binds and sticks while everything else just washes right through.
And then, to get it off, you add a high concentration of free glucose into the buffer. The free glucose competes with the immobilized glucose, releasing the concanavalin A with incredibly high specificity and purity.
Now, moving beyond purification, we need to detect and visualize these proteins. And that's where the exquisite specificity of antibodies comes into play.
We differentiate between two types. Polyclonal antibodies are a heterogeneous mix, recognizing several different binding sites, or epitopes, on an antigen.
But monoclonal antibodies
Are all identical. They recognize only a single specific epitope. And this homogeneity is critical because it yields much more reliable and specific results in analytical applications, especially for quantification.
Speaking of which, one of the most common applications for measuring protein concentration is the ELISA, the enzyme-linked immunosorbent assay.
The ELISA is a powerhouse of sensitivity. You link an enzyme to an antibody. If the target protein is present, the antibody-enzyme complex binds, and when you add a colorless substrate, the enzyme generates a colored product.
So it amplifies the signal.
Massively. This allows the technique to detect a less than a nanogram of a specific target protein in a really complex sample.
We can also visualize proteins in real time within a living cell, which is where things get really dynamic. Green fluorescent protein, GFP, is a perfect example.
Yes. By genetically fusing the gene for GFP to a protein of interest like the mineralocorticoid steroid receptor, researchers can visually track the protein's location.
And what did they find?
They discovered that the receptor just sits in the cytoplasm until the hormone is added. Upon binding the hormone, the fluorescently tagged receptor translocates immediately and visibly right into the nucleus.
Confirming its role as a transcription factor ready to control gene expression.
Exactly.
Finally, determining the precise sequence and 3D structure requires our most advanced analytical tools. Mass spectrometry has become central to proteomics.
It offers a highly accurate determination of mass, acting as a kind of molecular name tag based on a protein's composition.
But the real revolution is in sequencing, right?
Yeah.
Tandem.
Right. Tandem MS takes peptides, fragments them into smaller pieces in a vacuum, and then measures the masses of those fragments. The mass differences between the resulting peaks directly correspond to the mass of the individual amino acids lost,
So you can sequence the original peptide?
with high confidence. And this capability allows for rapid identification of proteins in complex mixtures using peptide mass fingerprinting.
Okay.
If you know the mass of all the fragments from a cleaved protein, you can often match that fingerprint to known sequences stored in genome databases, effectively identifying the protein without having to sequence every single residue.
But to get the full 3D blueprint, we rely on X-ray crystallography.
This is the most powerful method for high-resolution structure determination. You bombard a crystal of the protein with X-rays, the electrons in the atoms scatter the X-rays, and the recombination of those scattered waves gives you a complex diffraction pattern,
Which you then process mathematically.
using a Fourier transform to generate an electron density map. Biochemists then use this map to computationally build the atomic model of the protein, tracing where the electrons, and therefore the atoms, must be.
But what if you want to know how the protein behaves in its native environment, in solution?
Then you turn to nuclear magnetic resonance, or NMR spectroscopy. NMR reveals atomic structure in solution, reflecting the protein's natural conformational range, not just a static crystal snapshot.
NMR relies on two key pieces of information. First, chemical shifts.
Which tell us about the magnetic environment around the hydrogen nuclei.
And second, and more importantly for structure, the nuclear Overhauser effect, or NOE.
The NOE provides distance information. A no-ASY spectrum shows these cross peaks that identify pairs of protons that are physically close together, less than about 5 angstroms in the 3D structure.
Even if those protons are far apart in the linear amino acid sequence.
Exactly. By mapping all of these short-range distances, we can calculate the 3D shape of the molecule.
We shift now from the protein machinery back to the master blueprint, the nucleic acids, DNA and RNA.
Right. Long, linear polymers that carry all the information, built from 4 types of nucleotide building blocks. The double helix structure of DNA is absolutely fundamental to its function.
And that structure dictates a very specific method of copying, semi-conservative replication.
For decades, this was just a theory. The landmark Mendelssohn-Stahl experiment provided the definitive proof.
What did they do?
They labeled DNA with heavy nitrogen, N15, and then allowed the cells to replicate in a medium containing light nitrogen, N14.
So after one generation, what did they see?
After one generation, all the DNA was of an intermediate hybrid density.
Which ruled out conservative replication, where the parent strands would have stayed together.
It did. And when they let that hybrid DNA replicate for a second generation, they found 2 distinct bands. Half the DNA was the intermediate hybrid density, and half was light density.
Confirming that each daughter double helix contains one parent strand and one newly synthesized strand.
Yeah.
This replication process requires the strands to separate or melt, which is temperature dependent. In the lab, we can reversibly melt and reanneal, or hybridize, DNA.
And melting results in what we call hypochromism, which is an increased absorption of light at 260 nanometers.
Which is a useful assay, right?
A very important one. We use this property to monitor the stability of the double helix and, more importantly, to test the degree of sequence similarity between organisms.
How does that work?
If you mix the melted single strands from 2 species, how quickly and completely they reanneal tells you how complementary their sequences are.
The enzyme responsible for DNA synthesis is DNA polymerase.
A template-directed enzyme that synthesizes new DNA strictly in the 5' to 3' direction. And critically,
It needs a primer.
it absolutely requires a primer, a short segment of nucleic acid with a free 3' hydroxyl group to start its job. Without that primer, it's just stalled.
Now, let's look at the flow of information, the central dogma. Information flows from DNA to RNA,
Transcription.
and then to protein.
Translation. The synthesis of RNA is called transcription, and it's catalyzed by RNA polymerase.
What's the major functional difference between the two polymerases?
RNA polymerase also synthesizes in the 5' to 3' direction, but unlike its DNA counterpart, it does not require a primer.
Why the difference?
It's linked to fidelity. DNA replication needs extremely high fidelity and error checking, which is facilitated by the primer requirement and the subsequent proofreading activity. RNA synthesis is more permissive.
But the central dogma has famous exceptions, retroviruses like HIV.
Their genetic information flows backward, RNA to DNA.
Wow.
This is achieved by the viral enzyme reverse transcriptase. This enzyme is molecularly complex. It acts as both a DNA polymerase, copying the viral RNA genome into a complementary DNA strand,
Okay.
and an RNase H enzyme, which digests the original RNA template. The result is a double helical DNA version of the viral genome that can then be permanently incorporated into the host chromosome.
Once RNA is synthesized, the information has to be decoded via the genetic code.
Right. And we know it's degenerate. Most amino acids have more than one codon that specifies them, and it's nearly universal.
Why is that degeneracy important?
It's crucial for robustness. It means that many point mutations, especially in the third base of a codon, will still code for the same amino acid. It's an evolutionary buffer that allows life to tolerate a greater number of mutations without a functional consequence.
In eukaryotes, transcription is only the first step. Eukaryotic genes are often split by non-coding regions called introns, which have to be removed.
In a process called splicing.
Right.
And splicing is a fantastically complex operation. It's carried out by these large assemblies of proteins and small RNA molecules known as spliceosomes. The precision has to be staggering.
Because if the cut is off by just one base pair, the entire reading frame for the downstream protein is ruined.
Totally ruined.
So how does the spliceosome know exactly where to cut?
It relies on consensus sequences. Introns nearly always begin with the dinucleotide GU and end with AG, preceded by a pyrimidine-rich tract.
And that's the signal.
That sequence acts as the consensus signal that spliceosomes recognize to accurately remove the intron and join the protein coding regions, the exons, together. It's a crucial regulatory principle. The signal is simple, but the machinery is complex and highly specific. The ability to manipulate and analyze DNA fundamentally transformed biology. It moved it from a purely observational science to a synthetic one.
And the foundation of this revolution is the use of restriction enzymes.
Bacterial enzymes. They evolved to protect bacteria from viral invasion by chopping up foreign DNA.
Exactly. They recognize specific DNA sequences, which are usually inverted repeats or palindromes.
Sequences that read the same forwards and backward on opposing strands. And they make incredibly precise symmetrical cleavages within or near that recognition site.
And these precise cuts are what allow us to knit DNA fragments back together to build new molecules.
Yes. Many restriction enzymes make staggered cuts, creating complementary single-stranded ends, famously known as sticky ends.
Or cohesive ends.
Right. And because they're complementary, they have a specific affinity for each other, allowing fragments from different sources to spontaneously hybridize and be sealed permanently by DNA ligase.
Once we have fragments, we need to separate them. This is done with gel electrophoresis.
Right. DNA fragments are separated purely by size, with smaller fragments migrating faster through the gel matrix. We use agarose gels for larger fragments or polyacrylamide for smaller ones,
And then you stain them to see them.
with a fluorescent dye like ethidium bromide.
But if we need to find one specific sequence out of the millions in a complex genome, we use southern blotting.
Southern blotting combines separation and specific recognition. First, the complex mixture of DNA fragments is separated by electrophoresis. Then the fragments are denatured made single-stranded and transferred to a membrane.
And then you probe it.
Finally, the membrane is probed or hybridized with a labeled complementary DNA probe, allowing you to identify only the fragment containing the sequence you're interested in.
Now for the modern amplification powerhouse, PCR, the polymerase chain reaction.
This technique is genius in its simplicity and revolutionary in its application. It allows for the billion-fold amplification of a specific DNA segment in a test tube.
What do you need for it?
It relies on 4 key components: the target DNA, 2 synthetic primers that define the target region, the 4 DNA building blocks, and critically, the thermostable enzyme Taq DNA polymerase,
Which was isolated from a bacterium living in hot springs.
Thermus aquaticus.
And the genius of using Taq is that the process requires repeated cycles of heating the solution up to 95 degrees Celsius to separate the strands.
If you used a normal enzyme, you'd have to add new enzyme every single cycle.
The exquisite sensitivity of PCR is what makes it central to forensics.
Absolutely. Because it only requires a minute amount of starting material, the root of a single shed hair, a drop of saliva, a single cell. It can be used for DNA typing, allowing for unprecedented identification, disease diagnosis, and parentage testing.
Sequencing itself has gone through a radical shift. The classic Sanger sequencing, the dideoxy method, uses special analogs called ddNTPs.
Right. They lack the 3' hydroxyl group. When a ddNTP is incorporated, synthesis just stops.
So by running the resulting fragments, each terminated at a specific base, you can determine the sequence length based on the last base incorporated.
It was effective, but slow and expensive for massive genomes.
And the leap from Sanger to modern methods is staggering. The current standard is next-generation sequencing, or NGS.
NGS is defined by being highly parallel. Instead of sequencing one fragment at a time, these methods can acquire sequences from 1 million to 1 billion fragments simultaneously.
Oh.
One method, the reversible terminator method, uses nucleotides tagged with unique fluorescent labels and a reversibly blocked 3' end.
That's clever. The reversible block ensures that only one base is added per cycle before you image it to determine its identity.
And then you chemically deblock the end for the next round of synthesis. This combination of parallelism and chemistry allows for massive data throughput.
All these tools converge in recombinant DNA technology, which is the synthetic science of constructing new combinations of unrelated genes.
You take DNA fragments and insert them into vectors, plasmids, or viral DNA using restriction enzymes to create matching sticky ends and then seal them with DNA ligas.
These vectors allow the DNA to be amplified or cloned within host cells. And this technology, perfected in the 70s, paved the way for the genome projects. The finished sequence of the human genome revealed about 3 billion base pairs.
And here's the shocking insight. Despite our massive structural complexity, the human genome only contains about 20,000 to 25,000 protein-encoding genes.
Which is barely more than a simple roundworm.
Right.
So if the gene count isn't the measure of complexity, what is?
The complexity arises because many of those 25,000 genes encode multiple proteins via alternative splicing, where introns and exons can be rearranged to form different mRNAs and post-translational modifications.
And all the non-coding DNA.
A massive amount of the genome is non-coding DNA, including large numbers of mobile genetic elements, like the ALU sequence and lines, which have been inserting and duplicating themselves throughout evolutionary history. The sheer volume of DNA and protein sequence data we now have allows us to turn biochemistry into a historical science. We can trace evolution directly through these molecular footprints.
And proteins that descended from a common ancestor are called homologs.
Right.
We can detect these homologs through sequence comparison. But how do you find the connection when the sequences have diverged over millions or billions of years? I mean, simple identity checks won't cut it.
You're right. Simply looking for identical residues is insufficient for distantly related sequences. We rely on statistical alignment tools like scoring matrices. The Bluesum 62 matrix is the classic example.
Okay, what's the logic behind Bluesum 62?
It assigns higher scores to substitutions between biochemically similar residues, like substituting valine for leucine, than to radically different ones.
Because similar substitutions are much more likely to maintain protein function over evolutionary time.
Right. This allows the computational tools to detect homology with statistical confidence, even in sequences that share less than 20% identity.
That statistical analysis is powerful. The distant homology between hemoglobin, an oxygen-binding protein in plant roots, and human myoglobin is a textbook example.
On the surface, they look completely different, but the sophisticated scoring confirms they share a common ancestor, suggesting an ancient link between animal and plant oxygen transport systems.
This leads to a crucial insight derived from these comparisons.
Structural similarity persists longer in evolution than sequence similarity.
What does that mean?
It means if the sequence has diverged so much that homology is almost lost at the primary structure level, the overall three-dimensional shade of mechanism may still be recognizably similar. It suggests that folding scaffolds are highly conserved, even when the amino acid identity changes dramatically.
By tracing sequence differences and aligning these changes, we can construct evolutionary trees.
Estimating divergence times. This molecular approach provided the best timing estimates for the gene duplication events that separated myoglobin from the hemoglobin subunits and the alpha from the beta hemoglobin subunits.
And evolution is no longer just inference. It's an observable process. Modern techniques allow us to explore evolution experimentally.
For instance, PCR allows us to sequence ancient DNA, which helped place Neanderthals and Denisovans on the human evolutionary tree, showing they branched off prior to modern Homo sapiens.
And we can simulate early molecular evolution in the lab using a technique called helix.
Systematic evolution of ligands by exponential enrichment. helix starts with a vast randomized pool of nucleic acid sequences, up to 10 to the 15th different molecules. It then uses iterative cycles of selection and amplification.
Can you walk us through the selection part?
Sure. In a classic experiment, they selected for RNA molecules capable of binding to ATP. They passed the pool over a column where ATP was immobilized.
So only the RNA molecules that physically bound to ATP were retained.
Exactly. Then those retained RNA molecules are eluted, copied back into DNA using reverse transcriptase, amplified using PCR, and then transcribed back into RNA for the next round of selection.
And after several cycles.
The resulting RNA sequences showed a strong, specific affinity for ATP. This experiment simulates the molecular evolution that might have occurred early in life, where self-replicating RNA molecules evolved specific functions before complex protein enzymes emerged.
Moving from molecular history to dynamic molecular function, we turn to the classic system of oxygen transport, hemoglobin.
This system perfectly illustrates how structure facilitates complex, dynamic, and regulated function in the body.
We have to first compare myoglobin and hemoglobin.
Right. Myoglobin is a single polypeptide chain found in muscle tissue, and its job is to store oxygen. It grabs it tightly and only releases it when oxygen levels are critically low.
Whereas hemoglobin is a tetramer, a complex of 4 chains found in the blood, specialized for transporting oxygen.
Between the lungs and the tissues.
Yeah.
And both use the heme group, where oxygen binds reversibly to a central iron atom.
The functional difference lies in structure. Because myoglobin is a single chain, it follows simple binding kinetics. But hemoglobin, because it's a tetramer of 4 interacting subunits, exhibits cooperative binding.
Which is the hallmark of allostery.
Allostery means other site.
The binding of oxygen at one site, the heme group, affects the structure and thus the affinity of the other 3 distant sites. When one subunit binds oxygen, it causes a precise, subtle conformational shift across the interfaces between the subunits.
And what's the consequence of this structural shift?
That shift increases the affinity of the remaining 3 subunits for oxygen. And this cooperativity is what allows hemoglobin to efficiently bind oxygen in the oxygen-rich environment of the lungs.
Stabilizing the R, or relaxed state, with high affinity.
And then efficiently release it in the oxygen-poor tissues, stabilizing the T, or tense state, with low affinity. This structural dynamic is what makes oxygen transport across a large gradient possible.
This key state to R state shift is one of the most important conformational changes in all of biochemistry. So let's look at the real-world consequence of a single, minute error in that structure: sickle cell anemia.
The ultimate case study in how biochemistry directly drives pathology.
What's the cause?
It's a single-point mutation in the beta chain of hemoglobin. A glutamate residue is replaced by a valine residue at position 6.
And glutamate is hydrophilic and negatively charged, so it's happy on the protein surface. But valine.
Valine is nonpolar and highly hydrophobic.
That seemingly tiny substitution exposes a hydrophobic patch on the surface of the beta chain. And what happens next is a devastating example of the hydrophobic effect gone wrong.
When hemoglobin S, or HbS, is in its deoxy form, the low-affinity T state that is found in oxygen-poor tissues, this exposed hydrophobic valine residue finds a complementary hydrophobic pocket on an adjacent HbS molecule.
And this binding initiates a chain reaction.
The deoxy HbS molecules begin to aggregate uncontrollably into these large fibrous structures, long, rigid polymers.
And those fibers physically distort the typically disc-shaped red blood cells into a stiff, characteristic sickle shape.
Exactly. And these sickled cells are unable to pass easily through small capillaries. They clog blood flow, which leads to tissue damage, painful crises, and severe anemia.
Crucially, oxygenated HbS remains functional and does not aggregate.
It's only the deoxy form, stabilized by the T state, that precipitates.
So a single non-polar amino acid substitution, when magnified by the cooperative T-state conformation, leads to a massive, life-threatening aggregation. It's a perfect illustration of structure causing dysfunction.
And in a fascinating evolutionary twist, carrying only one copy of the HbS gene provides enhanced resistance to malaria, a parasite prevalent in certain geographic regions. This selective pressure explains the gene's persistent prevalence despite its severe consequences.
We also look at thalassemia, another major class of hemoglobin disease.
This is caused not by a structural change, but by the loss or substantial reduction of a hemoglobin chain.
So in alpha thalassemia,
Insufficient alpha chains are produced. This leads to the formation of beta chain tetramers called hemoglobin H.
and what's wrong with HbH?
It's defective. It binds oxygen with very high affinity and critically shows no cooperativity. So it grabs oxygen in the lungs, but fails to efficiently release it in the tissues.
And in beta thalassemia,
Here you have an excess of alpha chains, And these unpaired alpha chains are highly insoluble, forming aggregates that precipitate inside immature red blood cells,
which causes the cells to be destroyed.
resulting in severe anemia. These examples show that the correct stoichiometry and the proper affinity curve are just as crucial as the correct amino acid sequence. If proteins are the workhorses, enzymes are the highly specialized, highly regulated tools.
They're proteins that catalyze specific chemical reactions with immense power and specificity.
Right. They allow reactions to proceed millions or even billions of times faster than their uncatalyzed rate.
And that specificity is key.
Yeah.
You gave the example of trypsin, which specifically cleaves peptide bonds, but only after lysine or arginine residues.
The secret lies in the active site. This is the specific region that binds substrates and contains the catalytic residues.
And the primary function of the active site isn't just to bind the substrate.
No, it's to promote the formation of the highly unstable transition state, thus lowering the activation energy of the reaction. Because proteins are flexible, the active site is dynamic, undergoing critical conformational changes upon binding.
The quantitative study of enzyme activity is governed by Michaelis-Menten kinetics. If we plot the initial reaction velocity against the substrate concentration, what story does that curve tell us?
It tells us the limits and characteristics of the enzyme. As substrate concentration increases, the velocity eventually levels off, reaching Vmax, the maximal velocity, which occurs when the enzyme is completely saturated with substrate.
It's reached its speed limit. So
Right.
Vmax tells us the maximum turnover rate. How do we measure the enzyme's interest in its substrate?
That's where the Michaelis constant, column, comes in.
Yep.
Column is defined as the substrate concentration required to reach half Vmax.
And it reflects affinity.
A low cumulative generally means high affinity, as it takes less substrate to saturate the enzyme.
And then there's the ultimate metric of speed, the turnover number, or kcat.
kcat is the number of substrate molecules converted into product per unit time by a single enzyme molecule when it is saturated. It allows us to compare the catalytic efficiencies of different enzymes.
Some enzymes are catalytically perfect.
Like catalase, their kcat is so high they are limited only by the rate at which they collide with their substrate.
Many enzymes, though, handle multiple substrates. How do these reactions differ?
They fall into two main classes. The first are sequential reactions, which require all substrates to bind to the enzyme before any product is released, forming a ternary complex.
If these can be ordered.
Like lactate dehydrogenase, where NADH must always bind first, or random, where the binding order doesn't matter.
And the second class.
The second is the double displacement or ping-pong reaction. The defining feature here is that one product is released before all substrates bind. This involves a transiently substituted enzyme intermediate where a part of the enzyme is modified temporarily.
Okay. Finally, we must consider inhibition, which is critical for natural regulation and for drug development.
Irreversible inhibitors bind tightly, often covalently, and dissociate very slowly, essentially knocking the enzyme out permanently.
Like penicillin.
Penicillin chemically modifies the bacterial enzyme transpeptidase, preventing cell wall synthesis. Aspirin also works this way, covalently modifying cyclooxygenase, inhibiting inflammation.
Then we have reversible competitive inhibition.
This is when the inhibitor resembles the substrate and competes directly for the active site. The good news here is that this inhibition can be overcome simply by massively increasing the substrate concentration.
Like statins for cholesterol.
Exactly. Statins are competitive inhibitors of a key enzyme in cholesterol synthesis. They closely resemble the natural intermediate but can't be processed, slowing down the whole pathway.
And finally, there's a hybrid form, affinity labels or suicide inhibitors.
These are highly specific, irreversible inhibitors. They mimic the substrate, binding specifically to the active site, but then they are processed by the enzyme, causing them to suddenly react covalently with an active site residue, permanently inactivating the enzyme.
So they trick the enzyme into inactivating itself.
That's a perfect way to put it.
We've established that the active site lowers the activation energy. Now we need to look at the specific chemical toolkits enzymes use to achieve that incredible speed.
Beyond the binding energy from the substrate fit, enzymes employ 4 core chemical strategies.
First up,
Covalent catalysis. A reactive nucleophile in the active site forms a temporary covalent bond with the substrate, stabilizing the transition state before hydrolysis releases the product.
second, general acid-base catalysis.
Where a molecule other than water acts as a proton donor or acceptor, often involving the imidazole side chain of a histidine residue, which has a pico-A near physiological pH. Metal ion catalysis, where metal ions like Mg2+ or Zinc2+ facilitate reactions by polarizing bonds or stabilizing charged intermediates. And fourth is the simple but powerful catalysis by proximity and orientation.
Which is just bringing substrates together in the optimal alignment.
Exactly.
Let's start with the gold standard, the serine proteases like chymotrypsin, which cleave peptide bonds.
The mechanism here was revealed by stopped flow kinetics, which measures the reaction rate in the first few milliseconds.
And what did that show?
It showed two distinct phases. First, a rapid phase called acylation, where the enzyme is modified and the first product is released, forming a covalent acyl enzyme intermediate. This is followed by a much slower phase called deacylation, where the intermediate is hydrolyzed by water to regenerate the free enzyme.
And the key structure that executes this is the catalytic triad.
A set of three conserved residues, serine, histidine, and aspartate.
And how does it work?
The triad works synergistically. Aspartate anchors the histidine, and histidine acts as a general base. It accepts a proton from the serine hydroxyl group.
Which enhances the nucleophilicity of serine's oxygen.
Dramatically, it allows it to attack the otherwise unreactive peptide bond, initiating the acylation phase.
And we can test the necessity of these residues using site-directed mutagenesis.
If you change a crucial residue, say replacing serine 195 with an inactive residue like alanine, you see a dramatic multi-thousand-fold reduction in activity, confirming the immense contribution of the triad.
Moving to a different strategy, consider carbonic anhydrase, which catalyzes the extremely rapid hydration of carbon dioxide into bicarbonate.
This enzyme is essential for gas transport in the blood, and it relies heavily on metal ion catalysis, specifically a Zinc2+ ion.
What does the zinc do?
It plays a central role in activating a bound water molecule by lowering its pKi from 15.7 to about 7. The activated, more acidic water molecule is ready to attack the CO2.
And there's more to it.
The histidine residue acts as a general base, shuttling protons away from the active site and back into the solvent. This proton shuttling prevents a buildup of protons that would stall the reaction, enabling the enzyme's extraordinarily rapid turnover rate.
Our final catalytic strategy example involves restriction endonucleases, which cleave the phosphodiester backbone of DNA.
These enzymes absolutely require Mg2+ for activity. Just like carbonic anhydrase, the metal ion activates a water molecule, positioning it optimally for nucleophilic attack at the phosphorus atom of the backbone.
The mechanism of this hydrolysis was beautifully confirmed by stereochemistry experiments. What did they show?
They showed that cleavage results in an inversion of stereochemistry at the phosphorus atom. This is consistent only with a direct attack by water, an inline displacement, and it rules out any covalently bound intermediate.
So the mechanism is precise.
Mg2+ positions the water, and the water attacks the phosphorus, resulting in the hydrolysis of the bond and creating a free 3' hydroxyl group. If enzymes are the specialists, then regulation is the cellular traffic control system. One of the most common methods is allosteric control, typically seen in feedback inhibition.
This is where the final product of a metabolic pathway circles back to control the flux by inhibiting the enzyme that catalyzes the committed first step.
It's an elegant, self-regulating circuit. The classic example is cytidine triphosphate, or CTP, inhibiting aspartate transcarboamylase, or ATCase.
Okay.
CTP is the final product, and ATCase is the enzyme of the committed step in pyrimidine synthesis. CTP binds to the regulatory subunits of ATCase and stabilizes the T state, the inactive conformation, thereby slowing down the entire pathway when the product is abundant.
This behavior is explained by the concerted model, the MWC model.
Right. This model posits that the enzyme exists in two main states, T, tensine active, and R, relaxed active, which are in equilibrium. Substrate binding shifts the equilibrium toward the active R state, while allosteric inhibitors like CTP shift it toward the inactive T state.
Regulation also occurs via isozymes, or isoenzymes.
These are homologous enzymes that catalyze the same reaction but differ in their kinetic or regulatory properties, allowing them to adapt to specific tissue needs.
A good example is lactate dehydrogenase, LDH.
LDH exists as the M4 isozyme in skeletal muscle, which is adapted for an anaerobic environment and rapid ATP production. Conversely, the H4 isozyme found in the heart is adapted for the highly aerobic environment of cardiac muscle. Their slightly different kinetic properties ensure they function optimally under vastly different oxygen conditions.
Moving to perhaps the most widespread regulatory mechanism, reversible covalent modification.
With phosphorylation being the most common, ATP donates its terminal phosphoryl group, usually to the hydroxyl groups of serine, threonine, or tyrosine residues.
And this is a push-pull system. Kinases add the phosphate group, and phosphatases remove it.
Right. And phosphorylation often causes a massive conformational change due to the introduction of two negative charges, radically altering the protein's activity state.
The structure of the regulatory machinery is highly conserved. Protein kinase A, pKa, is a great example.
Its core catalytic domain is built on a conserved structure known as the kinase fold, and pKa activity is regulated by domain closure. The two lobes of the catalytic subunit move closer together upon substrate binding. Regulatory mechanisms like the binding of a regulatory subunit restrict this movement, ensuring the kinase is only active when and where the cell needs it.
Finally, we have irreversible regulation through zymogen activation.
Enzymes synthesized as inactive precursors, or zymogens, that are only activated when needed by a precise, irreversible proteolytic cleavage. This is essential for processes like digestion and blood clotting, which must be tightly controlled.
The blood clotting cascade is a stunning example of amplification and irreversible control.
Prothrombin is cleaved by proteolysis to form active thrombin. Thrombin, in turn, cleaves fibrinogen, a massive protein, into fibrin, which polymerizes to form the soft clot.
That cascade means a tiny initial signal is amplified by sequential enzyme activation into a massive, rapid physiological response.
And regulatory inhibitors ensure the system doesn't run rampant. For example, antithrombin III controls clotting. And in the lungs, alpha-1 antitrypsin protects lung tissue by inhibiting the digestive enzyme elastase.
There's a pathological link there.
Oxidation of a crucial methionine residue in alpha-1 antitrypsin, often caused by cigarette smoke, inactivates the inhibitor. Without that protection, elastase digests the lung tissue, leading to the development of emphysema.
We tend to focus heavily on nucleic acids and proteins, but carbohydrates, or glycans, are often overlooked as information-rich molecules.
They're central to cell survival and communication. And carbohydrate structure is deceptively complex. Monosaccharides, the basic building blocks, exhibit structural complexity through isomerism. When they cyclize, they create an additional asymmetric center called the anomeric carbon.
This results in anomers, like alpha-D-glucopyranose and beta-D-glucopyranose, which differ only in the orientation of the hydroxyl group at C1.
And that small difference can radically change how the sugar is metabolized or recognized by enzymes.
Complex carbohydrates are formed when monosaccharides are linked by O-glycosidic bonds.
The disaccharide maltose, for instance, has an alpha-1-villia-4 glycosidic bond between two glucose residues. Oligosaccharides have a specific directionality defined by the reducing end, which is the free anomeric carbon capable of opening the ring structure.
Many of the dynamic functions of the cell surface depend on glycoproteins, proteins with carbohydrate groups attached.
In fact, up to 50% of the proteome consists of glycoproteins, highlighting the importance of this modification.
We distinguish three main classes.
Simple glycoproteins, where the carbohydrate is N-linked to asparagine or O-linked to serine threonine. Proteoglycans, which are proteins conjugated to massive, negatively charged glycosaminoglycan chains, like aggrecan in cartilage. And mucoproteins, rich in O-linked carbohydrates, often serving as lubricants.
The physiological relevance is often about stability and trafficking. The hormone erythropoietin, EPO, is 40% carbohydrate by weight.
Its glycosylation is absolutely essential for stability and bioactivity in the bloodstream. Unglycosylated EPO is rapidly cleared and rendered ineffective.
The machinery for this is located outside the cytosol.
Glycosylation occurs in the lumen of the ER and the Golgi complex. The Golgi acts as the cell's major sorting center, altering and elaborating the carbohydrate units before the proteins are sent to their final destination.
The synthesis of these complex structures relies on specific enzymes called glycosyltransferases.
They catalyze the formation of glycosidic bonds. They account for 1 to 2% of all gene products, underscoring the metabolic commitment to carbohydrate synthesis.
This specificity dictates cellular recognition, most famously seen in the ABO blood groups.
Right. Type A individuals have a glycosyltransferase that adds N-acetylgalactosamine to the O antigen. Type B individuals have a different transferase that adds galactose. The O phenotype results from an inactivating mutation, leaving the basic O antigen unmodified.
Finally, lectins are specialized proteins that recognize and bind specific carbohydrate structures on cell surfaces.
This interaction is critical for cell contact, adhesion, and trafficking. Even viruses hijack this system. Viral hemagglutinin on the influenza virus recognizes sialic acid residues on host cell surfaces, which is the necessary first step for the virus to initiate infection. The organization of life depends entirely on establishing boundaries, which brings us to lipids and cell membranes.
Lipids are defined primarily by their insolubility in water and are built from basic components like fatty acids, and the structure of those fatty acids dictates membrane properties.
We look at two key factors that enhance the fluidity and lower the melting point: short chain length and the presence of cis unsaturation,
Cis double bonds create permanent kinks in the fatty acid chains.
which prevents them from packing tightly together in a solid structure. Saturated fatty acids lacking these kinks pack tightly, resulting in higher melting points and more rigid structures.
Membrane lipids are fundamentally amphipathic. They have a polar head group that loves water and hydrophobic tails that shun it.
This includes phosphoglycerides, sphingolipids, and glycolipids. Cholesterol also plays a key role in animal membranes, maintaining fluidity over a wide temperature range.
This amphipathic nature is the cause of membrane structure.
In aqueous media, these molecules spontaneously form thermodynamically favored lipid bilayers, often closing into liposomes. This spontaneous assembly minimizes the exposure of the hydrophobic tails to water, satisfying the second law of thermodynamics by maximizing water entropy.
The dynamic functions of the membrane transport, communication, catalysis are largely carried out by membrane proteins.
We classify them structurally. Integral proteins are embedded within the bilayer, often traversing it completely. They require strong detergents to be solubilized.
And you can often predict these transmembrane regions.
Because they typically consist of hydrophobic alpha helices sequences of about 20 consecutive non-polar amino acids, are often diagnostic of a membrane-spanning region.
In contrast, peripheral proteins are bound loosely to the surface by weaker electrostatic interactions or hydrogen bonds.
And they can be easily removed without disrupting the membrane.
Membranes are not static. Lateral diffusion, the movement of lipids and proteins within the plane of the membrane, is rapid.
But transverse diffusion, or flip-flop from one phase to the other, is very slow, which is essential for preserving the functional asymmetry of the membrane.
For example,
The sugar residues on glycoproteins are almost exclusively restricted to the external surface of the plasma membrane.
eukaryotic cells have extensive internal membranes, requiring complex mechanisms for moving material. This is where receptor-mediated endocytosis comes in.
Endocytosis begins when a ligand binds to a specific receptor, initiating the formation of an invagination, often a clathrin-coated pit, which pinches off to form an endosome vesicle.
The internalization of iron provides a perfect example of how this process is regulated.
Iron-bound transferrin binds to its receptor. The complex is internalized, and the cell uses proton pumps to lower the endosome luminal pH to about 5.5.
And this lower pH causes the iron to dissociate from transferrin.
But the receptor-transferrin complex remains intact. The receptor is then recycled back to the surface. This entire mechanism allows for the specific and efficient uptake of iron while minimizing the use of new resources. Membranes are highly functional barriers that require complex transport systems. These fall into two main modes.
Passive transport, or facilitated diffusion, where solutes move down their concentration gradient.
And active transport, which requires energy to move solutes against their gradient.
Primary active transport is driven directly by ATP hydrolysis. This includes the major class of P-type ATPases, like the sodium-potassium ATPase and the circocalcium ATPase.
Their mechanism is defined by forming a critical transient phosphorylase intermediate by using ATP. The enzyme is phosphorylated, and the energy released by the cleavage of the high-energy phosphate bond is coupled to conformational changes that shift ion access from one side of the membrane to the other.
Another critical class is the ABC transporters, APP binding cassette, which include the problematic multidrug resistance protein.
ABC transporters utilize the binding and hydrolysis of ATP at their ABC domains to drive massive conformational changes that open the channel to the opposite face of the membrane. This mechanism allows them to effectively export toxins or drugs out of the cell against immense concentration gradients.
Which is why cancer cells can become resistant to chemotherapy.
Exactly.
Then there is secondary active transport, which doesn't use ATP directly.
Instead, it exploits the pre-existing gradient of one ion, usually a proton or sodium, to power the transport of a second molecule against its own gradient.
Lactose permise in E. coli is the archetype for this.
It's a symporter that harnesses the energy of a proton gradient moving down its gradient to drive the thermodynamically unfavorable uptake of lactose against its gradient.
Now for the fastest transport mode channels. These provide a rapid, continuous passage for ions down their electrochemical gradient.
And their function is defined by selectivity, particularly evident in the potassium channel.
The K+ channel selectivity filter is a masterpiece of molecular design.
It acts like a molecular bouncer, built just for potassium. As the potassium ion enters, it sheds its shell of hydrating water molecules. The channel's carbonyl oxygens are perfectly spaced to accommodate and coordinate the larger K+ ion, replacing the energy lost from water desolvation.
But why is sodium rejected? Sodium is smaller than potassium.
It's rejected because it is too small to interact properly with the carbonyl oxygens in the selectivity filter. The energy lost when sodium is stripped of its water hydration shell cannot be fully compensated for inside the filter, making its passage thermodynamically unfavorable.
K+ and A+ channels are also voltage-gated.
They change conformation in response to the membrane potential. They contain segments that form domains known as paddles that are highly sensitive to the electrical environment. These paddles move in response to voltage changes to mechanically open or close the central pore.
And once open, channels spontaneously close very quickly through inactivation. This is explained by the classic ball and chain model.
The N-terminal domain of the channel acts as a positively charged ball tethered to the channel via a flexible chain. Once the channel pore opens, the ball is pulled into the opening, physically plugging the pore and quickly inactivating the channel.
Finally, we have cell-to-cell communication via gap junctions.
These form channels between adjacent cells built from two opposing connexons, each built from 6 connexin subunits,
And they're critical for linking cytoplasms.
allowing small molecules and ions to pass directly between cells. Importantly, they're regulated and closed by high internal calcium concentration or low pH. This mechanism protects healthy neighbors by sealing off injured or dying cells, preventing the spread of harmful signals. The coordination of these molecular events across a cell requires complex communication via signal-transduction pathways,
These pathways exhibit principles like amplification
where a small signal generates a large response,
and modularity.
where proteins use reusable domains to connect different pathways.
Let's start with the most common system, GPCRs, or G-protein coupled receptors.
The GPCR is a 7-TM, a 7-transmembrane helix receptor. When the primary messenger, like epinephrine, binds outside the cell, it causes a conformational change that activates the associated G protein on the inside of the cell.
How does that activation work?
The binding triggers the G-alpha subunit to exchange its bound GDP for GTP. The activated G-alpha S-GTP subunit then dissociates and moves to bind to adenylate cyclase.
And that's where the massive amplification begins.
Yes. G-alpha S stimulates adenylate cyclase, which converts ATP into the secondary messenger, CMP. A single-bound hormone molecule can activate hundreds of G proteins, each generating thousands of CANMP molecules. CAMP then activates protein kinase A, pKa, continuing the cascade downstream.
Another crucial pathway utilizes receptor tyrosine kinases, RTKs, exemplified by the insulin receptor.
Insulin binding causes receptor dimerization. The two receptor halves move close together, which leads to cross-phosphorylation of tyrosine residues on the partner subunit. This phosphorylation is the switch that activates the receptor's internal kinase domain.
And once activated, these phosphorylated tyrosines serve as molecular docking platforms. This is the definition of modularity in signaling.
Exactly. The phosphorylated tyrosines are specifically recognized by domains called SH2 domains found on downstream signaling proteins. The SH2 domains tether these downstream components to the membrane, ready to receive further instructions. The cell uses these modular domains, like SH2, to plug together different signaling components like molecular Lego blocks.
Unfortunately, defects in these finely tuned signaling pathways are strongly linked to diseases like cancer. Genes that are mutated to be constitutively active are called oncogenes.
The overexpression or mutation of the epidermal growth factor receptor, EGFR, is a classic example. Even without the ligand, the receptor may dimerize slightly, sending an inappropriate constant grow and divide signal.
And understanding this defect has led to some of the most successful targeted therapies.
Like Herceptin and Gleevec. Monoclonal antibodies like Herceptin target and block the HER2 receptor, reducing the growth signal. Similarly, the small molecule kinase inhibitor Gleevec targets the variable fusion kinase in chronic myelogenous leukemia, binding to its active site and shutting down the unregulated growth signal.
The entire field of modern drug targeting is built on understanding these molecular defects.
It is, and a devastating example of pathway hijacking is cholera toxin.
Right.
Cholera toxin chemically modifies the G-alpha subunit, the one that stimulates adenylate cyclase. The modification prevents the subunit from hydrolyzing its bound GTP back to GDP,
It traps the subunit in the active GTP-bound state.
meaning the system is permanently stuck in the on position.
And a result.
Persistent stimulation of adenylate cyclase leads to runaway CAMP production. This massive buildup of CAMP causes the intestine to secrete huge amounts of fluid and electrolytes, leading to massive, persistent diarrhea and dehydration.
That brings us to the end of this incredibly dense deep dive. What a journey through the architectural and functional principles that govern life, from single bonds to complex signal amplification.
We've established a clear chain of command and regulation. Life relies on information encoded in DNA, executed by modular proteins, precisely regulated by kinetics, allosteric, and phosphorylation,
All organized by dynamic membranes.
and coordinated by complex amplified signal networks. The most fundamental idea is that molecular structure dictates function, and regulatory principles ensure stability and responsiveness across all scales.
So what does this all mean for the bigger picture? We've seen how even the most complex processes, like signal-transduction and allosteric oxygen transport, rely on molecular structures that existed billions of years ago.
Right.
And we discussed the metamorphic lymphotactin protein, which can switch its structure and function entirely based on need.
That raises a final provocative question for you to consider as you continue to explore this field. We currently assume that most proteins have a single fixed structure, but given that molecular shape shifting is clearly possible, as demonstrated by lymphotactin and even prion proteins, how many proteins in the human body do we currently think have a single fixed structure but are actually metamorphic?
Waiting for a subtle signal.
Like a binding partner, a metal ion, or a change in pH to completely redefine their biological role. Understanding that flexibility may be the key to unlocking the next generation of therapeutic targets.
Something to think about, indeed. Thank you for joining us for this deep dive into the molecular structure, information flow, and cellular dynamics that define life. We'll catch you next time.